Hello, welcome to this lecture on biomathematics. Today we will discuss and compute certain quantities for DNA and understand how do we apply statistical thermodynamics, ideas from thermodynamics to understand various properties of DNA. So, today we are again continuing the same theme that we have been discussing, same module that I have been discussing, basically statistical thermodynamics of biological systems. And what we will be computing at the beginning is partition function for double stranded DNA. So, there is this model for DNA known as worm like chain model, and so this worm like chain model, let me write it here, worm like chain model. In short, this is known as WLC. This model is DNA is thought of as a line, inextensible line with a bending energy. The only energy is bending energy, and the discrete version of this is thinking of DNA as a set of vectors. So, this is tangent vectors T1, T2, T3 and so on and so forth. There will be some similar n tangent vectors. So, up to there will be like T n, there will be n tangent vectors, and this orientation of this tangent vectors in 3D will decide the configuration of the DNA. So, now the energy of such a system is given by in a certain manner. So, but just before that, the orientation of this in 3D, whatever way this each tangent vector orients will decide the orientation of the DNA. And one of the properties is that the DNA wouldn't like to bend. There is an energy cost to bend the DNA. To make a loop out of DNA, there is a cost of, you have to spend some energy, and that energy is basically the bending energy, which is given by some constant A, which is called the bending stiffness, times sum over all tangent vectors I is equal to 1 to n, there are n tangent vectors, 1 minus T I dot T I plus 1. So, this is basically the bending energy of a DNA. Sum over I 1 minus T I dot T I plus 1. So, if there are if the DNA of like 3 base pair, this will be A into 1 minus T 1 dot T 2 plus A into 1 minus T 2 dot T 3. Okay, there are 3 tangent vectors, only then this will look like this T 1, T 2 and T 3. Then if there are many, there will be plus dot 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 A into 1 minus T n minus 1 dot T n. So, this is the energy, the bending energy. This is the only interaction here is the bending interaction, and this is the bending energy of a double stranded DNA, which has n tangent vectors and each of these unit vectors, and the mode, the unit, each of this unit, this has a particular. So, mod T is 1, that means we take all these vectors unit vectors. So, now given this, we want to calculate the partition function, and we said that the partition function is basically sum over all states. So, that means you integrate over all possible dt 1, the first tangent vector can take any orientation. So, this is vector in 3D. So, it can take all orientation in 3D. If you have this vector and this vector, the first vector T 1 can orient in any way, with T 2 is fixed this particular way, and T 1, the green one, can orient any way you want. So, the integral d T 1 means you sum over all these orientations. Integral, the second one, integral d T 2, integral d T 3, so on and so forth, integral d T n e power minus beta E b, where beta is 1 over k b t. So, this is the partition function. So, this is the partition function Z for the double stranded DNA, which has an energy E b, and this partition function is given by this formula integral dt1. This means sum over all possible conformations e power minus beta e. So, this integrals here mean that sum over all possible conformations. First tangent can have all angles, all orientations. Second tangent can have all orientations. Third tangent can have all orientations, and nth tangent can have all orientations. So, each base pair, you can imagine each tangent as each base pair. So, each base pair can orient in any way, or this can be thought of as, in general, if you could imagine DNA as a polymer, each monomer can orient in any direction, and this into sum over all this will give you the partition function. Now, we said that what is dt1? dt1 is, you can think dt1 as a vector in 3D. So, the basically dt1 can have any orientation, means if you imagine one reference point, reference line, for example, if you take this as x axis and y axis, the vector which you are interested in can take any angle. So, this can be described by a set of angles theta and phi, just like we know from spherical polar coordinates. And since the length of the tangent vector is a constant, this dt1 is nothing but integral angle from 0 to 2 pi and integral over theta from 0 to pi. Okay, so, this is the integral, and this will have, sorry, there will be a sin theta here. So, this is sin theta d theta, sin theta e power minus beta e. So, as you might know that the volume element in spherical polar coordinates is basically d phi sin theta d theta with, so the only angle change happens, only phi angle can change, theta angle can change in this particular manner. Given that the length is constant and unit vector 1, this will be the, and the length does not change, this will be the volume element. So, that is why this integral is d phi and sin theta d theta. Now, sin theta d theta can be written as d of cos theta, sin theta d theta can be written as d of cos theta. So, you can write this of d of cos theta and then accordingly change the limits, and this dt1, if you do that, you will get that integral dt1 can be written as integral d phi. So, since there is dt1, let us also call this d phi 1, the first phi angle, d cos theta 1. So, this will be 0 to 2 pi, and this will be minus 1 to plus 1. If you change the limit appropriately, you will get that this is from minus 1 to plus 1. So, and then you have similarly, you have 0 to 2 pi d phi 2. So, this is dt 2, you can write integral dt 2 as d phi 2 minus 1 to plus 1 d cos theta 2. So, if you know dt1, dt2 like this, we can write everything like this. So, essentially the partition function will turn out to be, so partition function essentially is integral dt1, integral dt2, so on and so forth, integral dtn e power minus beta e, where this is the bending energy, and this can be written as integral d phi 1, integral d cos theta 1, integral d phi 2, integral d cos theta 2 dot dot dot, integral d phi n, integral d cos theta n e power minus beta E b. This is the bending energy. Now, all these limits are same. Here this limit is 0 to 2 pi, here it is minus 1 to 1. So, now what is E b? E b is sum over I, some constant a times 1 minus T I dot T I plus 1. T I dot T I plus 1 can be written as cos theta, because this is two unit vectors and the product is cos theta and the magnitude is 1, each has unit magnitude 1. So, this is just nothing but 1 minus cos theta. So, you can write this 1 minus cos theta here. So, this E b depends only on cos theta. So, this is nothing but a into sum over I 1 minus cos theta I. So, this depends only on cos theta. So, the whole integral does not depend on phi at all. So, because this function does not depend on phi at all, only theta it depends. So, we can take out all the phi integrals out and you can do all the phi integral. So, integral 0 to 2 pi d phi is just 2 pi. So, we can do this, and if you rewrite this, you can see that e power minus beta E bending energy can be written as e power minus beta sum over I a times 1 minus cos theta I. Now, if you take this sum, so this is basically e power minus beta E 1. So, if we call this part a into 1 minus cos theta as alpha I, a into 1 minus cos theta I, let us call it as alpha I. So, this is basically, this is nothing but e power minus beta sum over I alpha I. So, this is nothing but e power minus beta into alpha 1 plus alpha 2 plus alpha 3 dot dot dot. So, e power anything sum can be written as product. So, e power minus beta alpha 1 plus alpha 2 plus alpha 3 etcetera can be written as the product of this. So, if we rewrite this, we will get e power minus beta alpha 1 dot e power minus beta alpha 2 dot dot dot. So, this is the sum can be written. So, this sum can be written as a product of alpha n. So, essentially the partition function can be written as, so this is product of n such terms. So, it turns out that you can think about it and you will see that the partition function is essentially integral d phi, integral d cos theta, d phi goes from 0 to 2 pi, d cos theta going limit minus 1 to plus 1, e power minus beta a into 1 minus cos theta whole power n. So, there are n such terms and there is a product of n such terms, and that will give you the partition function. So, if you calculate just this two integrals and find the product of this n times, you will get the partition function for a double standard DNA. So, this is very easy to do. This is 2 pi as we know, and we can easily calculate this integral. So, here itself e power minus beta, this a term will come out. So, that e power minus beta a into 1, this term will come out, and we can call cos theta as x, let us call cos theta as x. So, if we rewrite this a bit, what do we get? Let us quickly rewrite this. If we rewrite whatever is seen here, what do we get? So, let us rewrite these terms. If we rewrite this, this is basically 2 pi. So, there will be 2 pi power n. because integral 0 to pi, this is 2 pi. So, and there is n whole power n. So, there is 2 pi power n, and there is e power minus beta a n times. So, there is e power minus beta a n times. So, this is e power minus beta a n times, and then there is this integral a cos theta, let me call this s, minus 1 to 1 d cos theta as dx, and there is e power minus beta a into minus cos theta, this minus minus becomes plus. So, this e power minus minus becomes plus beta a x. So, this is the integral we have to do, integral dx minus 1 to 1 e power beta a x. This is easy to do. So, this is basically 2 pi whole power n e power minus beta a n, and this is integral of e power beta a x dx is basically e power beta a x divided by beta a in the limits minus 1 to 1. So, if you apply the limits, this will become minus and minus. So, sorry, there is a plus here. So, you can apply the limits. So, if you apply the limits, it will be e power beta a minus e power minus beta a. So, let us apply this limit properly. So, if we apply the limits, what do we get? So, if we do this and apply the limits, what you will end up getting is basically the partition function you will get as 2 pi whole power n e power minus beta a n. You will get sine hyperbolic beta a divided by beta a whole power n. So, this is the partition function you will get, and log of z minus k T log z will give you the free energy. So, the free energy of a double stranded DNA F is minus k B T log z. So, from this we can calculate the partition function and the free energy. Now, this is the answer. This is for the partition function for a double stranded DNA is 2 pi power n e power minus beta a n sine hyperbolic beta a by beta a whole power n. So, it depends on this quantity called A, which is the bending stiffness of the DNA. It depends on k beta, beta is basically 1 over k B T. So, it depends on the temperature. So, it only depends on three things. It depends on 3 parameters. So, let us note this parameter. It depends on this n. It depends on beta, which is 1 over k B T, which is temperature. It depends on A, which is the bending stiffness. So, there are 3 parameters. The property of the DNA will depend on 3 parameters. How many monomers are there? How many base pairs are there? N is basically the length related to the length of the DNA. It depends on the temperature, of course. It depends on A, which is the bending stiffness. So, all property, free energy will give you all properties of the DNA. You can derive all properties of the DNA from free energy, and we will see some examples immediately. We will discuss for even simpler case of single stranded DNA, we will discuss what is the, how do we get free energy, and from free energy how do we get various measurable quantities. But at this moment what we learn is how to calculate free energy, and immediately we will learn how to get measurable quantities from the free energy. So, this is what we discuss now is how do we get free energy and partition function and free energy for double stranded DNA. Now, what we are going to learn is how do we get something useful from this. What is the use of knowing free energy? What is the use of knowing partition function? The answer is we can get all measurable statistical quantities from this. So, let us think of very simple example first, and then we will go to the case of DNA again, and then we will do a calculation and see that something which you didn't know till now can be estimated, can be calculated sitting here by knowing what we learned so far. very simple idea. If you know how to calculate partition function and free energy, we can calculate a few things that we didn't know so far related to DNA, for example, here, and any other system that you can think of. Okay, now let us think of first a simple spring like protein, which is our favorite example for describing or discussing any statistical thermodynamics idea, because this imagining a protein like a spring will help us thinking ahead, and then will help us to simplify a lot of things and have the basic have the important idea seen in the example. So, let us think of two atoms connected by a spring, just like we have been thinking, two atoms connected by a spring like interaction, and this is in water. So, this is water here. So, as we said, since due to the Brownian motion, this spring will be extended and compressed. So, this atoms will be doing, these molecules will be doing Brownian motion. So, if you imagine, they will be just doing this vibrating like this, because they will be hit by Brownian water molecules doing Brownian motion. So, essentially this atoms will be doing vibrating like this. Now, we can ask the question, what is the distance between these two, root mean square distance between these two? Like, will it be, how much it will fluctuate? How much this distance will fluctuate? This answer, this is given by this quantity, which is basically, this x square average minus x average square. This quantity is called the sigma square, which is basically the sigma, which is the sigma square is a variance, and sigma is the square root of this is a standard deviation. So, this will give you our x is, if x is the distance. So, x is basically this distance from here to here, this distance is x, and if we calculate x square average minus x average square, this will tell you how much this protein will fluctuate. So, how do we compute this from partition function? We can compute this, and that will tell us how much this will fluctuate and what will be the position of these atoms on an average. We will be able to calculate. So, let us take, so what is the energy of a spring? So, typically the energy of a spring is basically half k x square. So, energy of a spring like, so if you have a spring like this, the energy we know is half k x square. X is basically the distance from, so here let us take x as basically some, so let us call this x is basically the distance from equilibrium. So, at equilibrium, let us say you have such a spring, this can get stretched, then this distance we can call it x here. So, then how if it stretched by a distance of x, then the energy is half k x square. Even if it is compressed by a distance of x, the energy is half k x square. If you compress the spring by an amount x, this energy is half k x square. So, now what will be the partition function? The partition function will be z, will be e power minus beta, which is 1 over k b t, half k x square, this is e power minus beta e, and all possible x. So, the x can go have all possible values. So, integral over x, let me take minus infinity to infinity. That means, I can take this spring from all the way from minus infinity to the infinity throughout this. So, this way if you do this integral, the answer of this will give you the partition function. So, we will have a look at this once more. So, the partition function for a spring doing Brownian motion, the fluctuating spring, will be dx e power minus beta by 2 k x square. So, this will be the partition function, and if we calculate this, from this we can calculate free energy, and from that we can calculate various useful quantities. So, let us think about this a minute. Now, let us do this calculation. So, if you think about this is an something, so beta is a constant, k is a constant. So, this beta k by 2, I can call this. So, let me call this beta k by 2 as some new notation. Let me call this k tilde. So, k tilde is basically beta k by 2. So, this is e power minus k tilde x square. So, this is like Gaussian integral. So, this is like a Gaussian function, e power minus some constant into x square. So, integral of a Gaussian function we know. So, let us say that integral of a Gaussian function, integral e power minus beta k tilde x square minus infinity to infinity dx is our, and this answer is root of pi by k tilde. So, this is the answer. If such an integral, this is called a Gaussian function, and this is the function which looks like a normal distribution, something we discussed. So, this answer integral of this from minus infinity to infinity root of this. So, this is basically the answer. Now, we can ask the question, what is the probability of finding the spring at a particular distance x is equal to, let us say 20 unit, or you can ask what is the probability to find the DNA at a distance 35 units, and the answer will be this. So, the probability of finding the spring when x is equal to, let us say x is equal to x prime is some number, some particular value which could be 2 nanometer, 3 nanometer, 5 nanometer. So, this is e power minus beta half k x is equal to x prime square divided by root of pi by root of k tilde. Root of k tilde 1 over k tilde I can write as root here, and k tilde we wrote as beta k by 2. So, this will be the answer. What is the probability of finding the spring at a distance? If you substitute x prime is equal to 3 nanometer and the appropriate spring constant beta k by 2 x prime square, there is no extra k here. So, this is the energy, and if you substitute x prime, k and beta appropriately, you will get the probability. So, from this we can get the probability. Now, we can ask the question, okay, if you know the probability, we can calculate x square average. We said that the definition of x square average always is basically x square average is defined as integral x square p of x dx minus infinity to infinity. This is x square average. So, let us calculate x square average. X square average is equal to minus infinity to infinity. P of x will be e power minus beta k by 2 into x k by 2 into x square divided by root of pi by k tilde. So, now here you have root of pi by k tilde, and you have to multiply with x square and integrate over this. So, again let us call this part as k tilde beta k by 2. So, this integral will turn out to be, if we do this correctly, what do we, it is a very simple integral. So, what do we have? What we have is basically, what do we have here is integral minus infinity to infinity, that 1 over z, which is root of pi by k tilde I take out. So, this is root of k tilde by pi x square e power minus k tilde x square. This is the integral dx. This is the integral we have to do. So, it turns out that there is a trick to do this integral. There is an x square here, and if you take the derivative of this with respect to k tilde and I put a minus sign, you will get this. So, it turns out that just see this carefully and try to do this yourself. So, this is nothing but is equal to root of k tilde by root of phi into minus del by del k tilde. You do this yourself after seeing this and see what I am saying is correct or not. Del by del k tilde integral minus infinity to infinity e power minus k tilde x square dx. It turns out that if you take derivative with respect to k tilde and x square will come out, that x square is same as this x square. So, you can rewrite this in this particular manner, you can do this carefully. So, if you do this, you will get this answer, and we know that the answer for this is again a Gaussian integral. The answer for this is again root of pi by k tilde. Root of pi by k tilde is the answer for root of phi divided by k tilde is the answer for this. So, once we have this, we can calculate the derivative of this and then get this answer, which is basically this will give you x square average. This is what we are calculating, and x square average is this. So, let us do this slowly. So, this is very simple algebra, nothing complicated. So, if you think about it, so you have here root of k tilde by root of pi, and this is root of minus del by del k tilde or root of pi by k tilde. So, this is the answer, and root pi comes out, and what you have is basically if you do this, root pi and root pi cancels, and what you have is root of k tilde into del by del k tilde into there is 1 over root of k tilde, which is k tilde power half minus half. So, this will turn out to be, and there is a minus sign here from here. So, this minus half this, and so this will turn out to be root of k tilde into half minus half minus 1. So, basically what you have here is k power minus 3 by 2. So, k power minus half minus 1, which is k tilde power minus 3 by 2. So, basically this will turn out to be the answer, final answer will turn out to be x square average is equal to 1 over k tilde with a half sign. This is the x square average. What is k tilde? 1 over 2 into k tilde is beta, which is 1 over k b t. So, I can take k b t over here and k, and there is a by 2. So, half. So, there is a 2 here. So, this 2 and 2 cancels. So, this is basically k b t by k. So, it turns out that the x square, the average x square is k b t by k. The more the temperature, the more the x square average is, the more the spring constant, less the x square average is. So, it turns out that the x average will turn out to be 0. You can see this similar way, x average will turn out to be 0. So, the standard deviation is nothing but x square average. So, we have this same thing as standard deviation. So, we have x square average is equal to k b t by k. So, this is some interesting quantity that we can calculate. How much protein will fluctuate, because the whole fluctuation is because of the heating by water, and it is statistical. We have to calculate quantities that is statistical in nature, like x square average and the sigma, the standard deviation. Essentially, what we computed here, what the quantity here k b t by k physically tells you what is the meaning of this. It means that if you have a protein in water, it can fluctuate depending on the temperature it will fluctuate. So, it is k b t divided by k, which is basically the elastic k depends on the decides the elastic property of this protein. So, if depending on the elastic property of the protein, we can say that this k is the fluctuation will vary. Now, let us quickly think of a DNA. Now, and let us see what do we mean by such things we can calculate for DNA also. And let's think of a long chain in water. So, for a minute let us think of a single stranded DNA. So, a single stranded DNA, a long DNA can be thought of as a filament like this, and this is in water. There is water everywhere. The appropriate buffer, and think about this end and this end, and imagine that you are holding this end, you are fixing this end at some particular place and pulling the other end. So, as you pull this, you can imagine that the distance from this end to this end will keep increasing. So, imagine that you are pulling a rope which is like this slowly, very slowly, and if you imagine that you are plotting the distance from one end to the other end, how will the graph look like? So, let us think about this as how will the graph look like. So, the graph will look like, so let us say we are plotting this, the force we apply and pull it, and the distance from one end to the other end. So, let me call this r, distance from one end to the other end. So, it turns first the distance from one end to the other end is a constant, then it will slowly increase, and then finally, it will reach its maximum length. So, smooth, this is pretty smooth. So, this will look like this, but look at this particular region. Here this is almost like a linear part. In this region between this and this, it appears as if force is proportional to the distance. So, what is here is force is equal to some constant times r. So, this constant, this is like a spring, this is like a f is equal to k x is like a spring equation. So, in this regime, the DNA behaves, or any polymer for that matter would behave like a linear spring, like f is proportional to r. So, there is a regime where any protein will behave like a linear spring, and what is the reason for this? The reason for this, essentially think about this a minute, this reason for this is entropy. If you did not have any entropy, if they are at 0 temperature, the DNA would take, it wouldn't like to bend. So, it would take basically a very straight filament, it will be like only one configuration, but due to entropy it can go to various conformations, and this elasticity, this can be also, this is often called entropic elasticity. Now, imagine that we are doing this for a single stranded DNA, pulling a single stranded DNA with some force F, and we can calculate how much this k will be, how much this spring constant will be. So, this is just like, so imagine that for a moment, this is your, just like we discussed this example of a spring in water, exactly like similar way you can think of a DNA in water. It is a coiled DNA, and the distance between this and this end is equivalent to the distance between this end and this end of the DNA. So, if you got here a relation, we said x average square is k b t by k. Similarly, what will be the end to end distance square. What will be the R average square? It turns out that the R square average can be computed in a similar manner, and we can say that this is something which we discussed before, that if you had random orientation for each of this monomer, the distance from here to here, this distance let me call R, this is basically the distance from one end to this other end. If we call this R, this R square average will turn out to be B square, which is the length of this each subunit, times n. This will turn out to be this is something we discussed before. So, this will turn out to be R square is B square n, and we also just saw that for a spring we said that K, the R square average, if it is like a spring, will be equal to K B T by K. Now, equate this two, and it turns out that actually there is a, if you think of this only in one direction, fluctuation only in one direction, there will be only 1/3 of this in each direction. So, you can think this is only in one direction, then it will be 1/3 of this, but if you take off this whole thing, you will get, equate this to, you will get that K is nothing but K B T by B square n. So, this is in 3D. So, if you 1/3 of this, if you take, you will get a 3 here, because this is, so if you take R x square only in one direction. So, in the direction of, if we call this as x axis, x direction, we can put a 1/3 here. So, then this will turn out to be 3 K B T by B square. So, this can be thought of as, without doing much of calculation, we can think of, we can just equating a couple of things that we already know, we can guess how much will be the spring constant for a, if you think of it as a spring, the DNA that can be pulled like this, which will behave like a spring, the spring constant can be estimated here. We can calculate this exactly, but without doing much of calculation, we can estimate, we can guess what the answer will be. Now, what is the use of all this? Again, this all telling some property of the DNA. If the DNA is in a medium and the thermal behavior, due to this thermal motion, thermal Brownian motion, DNA will behave as if like a spring, and the spring constant can be estimated, and from this you can know how much force you need to pull. If you know the K, which is the spring constant, we can know how much force you need to pull the DNA and straighten them. This is, of course, what I said is for only for single stranded DNA. For double stranded DNA, this is much more complicated. Whatever I discussed in this part is for single stranded DNA, where you don't need energy to bend, and for double stranded DNA, things are more complicated. So, now we will do a detailed calculation later by applying real force to a single stranded DNA and calculate the partition function, and from that we can calculate something called this exact relation between force and R. So, we can calculate what will be this relation F versus R relation, which will look like this. We can calculate this full, we only estimated this part, but we can calculate this full part for small forces. For large forces, there will be a slightly different behavior, and we can calculate all this all this in detail. So, we will, this is also again simple statistical mechanics, statistical thermodynamics calculation. We will do that calculation, and we will estimate this pulling of DNA, and then the result from that we will think about this. So, basically what we have been doing so far is applying ideas from statistical thermodynamics to calculate partition function. Now, one more thing I should say, that we have calculated partition function and if we calculate partition function, we can get free energy, which is log of partition function, and it turns out that the derivative of the free energy will give you various quantities. So, if you calculate the free energy as a function of force or a function of some other quantity, if you take the derivative of the free energy, you will get various measurable quantities. So, it turns out that there are two types of free energy that you typically come across, 2 or 3. So, one is called Gibbs free energy, which is represented by G. So, Gibbs free energy G and Helmholtz free energy F. This Gibbs free energy is a function of, typically is a function of pressure and temperature, and this is a function of volume and temperature. This is the difference between these two. What does this mean is that if you fix the pressure and if you fix the temperature, and if you also fix, in the case of a total number in the system, which we are not discussing here, we always take it as fixed, we will get this free energy. If you do the experiment and by fixing the volume and temperature, you will calculate what you will be calculating in this F. Now, what is pressure? Pressure is basically force per unit area. So, pressure and force are equivalent. So, when you think of pulling DNA, pressure is like force. So, in the case of pulling DNA experiments, you can say that the G, if you take a DNA and fix one end, so you have a DNA and apply a force at the other end and ask the question, what is the free energy of this system? What you will be calculating is the Gibbs free energy, because you are calculating it at a given force and a given temperature. And it turns out that if you know this G, by taking the derivative del G by del F, you will be getting this end to end distance r along a particular axis. So, by doing del G by del F, you get this end to end distance. So, basically knowing the free energy, you will, by taking the derivative of free energy, we can get all measurable quantities. If you were to calculate do the experiment with fixed length, you fix the distance between the end to end of the DNA and calculate the free energy from that case and take the derivative with respect to length, you will get the equivalent of force. So, basically we can calculate free energy, and by finding the derivative of the free energy, we can calculate the various measurable quantities. So, think about a few things, like you might have heard of various things in thermodynamics. Let us list a set of things that you have heard in thermodynamics. So, you have heard pressure and volume, temperature, entropy, force, distance. So, all of this has a property that P into V has a dimension of energy, T into S has a dimension of energy, F into D has a dimension of energy. You can also say something called mu, which is chemical potential, N is the number, this also has dimension of, the product has a dimension of energy. So, there are two things here. See, there are two quantities, all this belong to one set of quantities and all this belong to another set of quantities, such that their product has a dimension of energy. So, this basically the PV is one pair, TS is one pair, FD is one pair, mu n is one pair. So, this is just like this is like forces, you can call this as generalized forces, and this is some kind of conjugate variable to this, but just enough to remember that there are many pairs in thermodynamics, pair 1, pair 2, pair 3, pair 4. Now, you can do any experiment by fixing any one quantity from each of these pairs. So, when you say some free energy G, we can fix basically pressure and force are same. So, there are only 3 pairs essentially. So, we can either fix F from this, T from this and N from this. This will give you some free energy. You can compute some other free energy, which is basically fix, instead of F you fix D, the distance or length. You can fix T and you can fix N. This will be another free energy, and you can think of many other free energies by fixing any one quantity from each pair. You can fix either F or D, mu or N, P or V is one of the, take 3 pairs, basically this pair and this pair are same. So, this pair is kind of redundant, because pressure is nothing but, force is nothing but pressure in one dimension. So, 3D there are only 3 pairs, 1, 2 and 3. So, we can fix one of this from each pair and calculate appropriate free energy. So, and by taking the derivative of the free energy with any of these quantities in the bracket, we can get the conjugate variable. If you take the derivative of free energy with respect to F, you will get the average distance. If you take calculate this free energy and take the derivative with respect to N, del F by del N, you will get mu. So, in this manner, we can think of computing various quantities and the measurable quantities. All these quantities can be computed by computing free energy. So, we will continue this discussion in the next class. So, for this moment we will stop here. So, what we discussed is basically a model, worm-like chain model for double standard DNA. Then we discussed how to calculate partition function for this. Then we discussed partition function for a spring in water, and we calculated the fluctuation, how much it will be fluctuating, and the spring constant for a single standard DNA, the effective spring constant if you were to pull this. So, with this I will stop today's lecture. We will continue in the next lecture. Bye.